what we finished with on Tuesday. So we started to talk about the membranes. Yes, the membranes, and we have talked about the serous membranes, right? And we talked about the mucous membranes, and we stopped at serous membranes. I'm trying to open the recorder. Okay, how about now? Awesome. So we talked about cutaneous, which is skin. We talked about mucous membranes, which is the lining of lumens, right? That's another word that I want you to know. We're gonna use this word quite often. Lumen. Do you understand what the lumen is? If you look down the tube, what you're looking at, that empty space, is called lumen. So there can be a lumen of intestine, lumen of the blood vessel, lumen of bronchi. The spelling for the word is lumen. Okay? I'm just going to use it quite often, I think. And so serous membranes, as we mentioned, they produce the watery fluid, the serous secretion, which lubricates internal organs. Prevents them from rubbing against each other. Now, there are several serous membranes in our body. They cover organs, but another thing that they cover are the cavities in which those organs are housed. For example, we have a membrane that is called pericardium, which lines the heart. But, as you may remember, heart is located in one of the cavities of the ventral aspect, in the pericardial cavity, right? In the pericardium. So if you think about it, if you have an organ in the cavity, every time you move, this organ will rub against even the walls of the cavity. So that serous membrane, the pericardium, lines both the organ and the walls of the cavity. Does that make sense? Now, interesting thing about this serous membrane, and about many others, is that you can technically say, okay, we have the pericardium, the layer that lines the organ, and the layer, sorry, it's not extremely precise, and the layer that lines the cavity. Layer that lines the organ will be called visceral. Layer that lines the cavity will be called parietal. So essentially you have two layers, right? But you know what's interesting? It is essentially the same membrane, which can be illustrated if you look at this balloon. So you have a balloon, okay? You take your fist and you stick it into the balloon. Your fist is covered, right, with sort of rubber, okay? And there's a space between the layer that covers your fist and the layer that is sort of an outside from it, okay? This is essentially would be visceral, and this would be parietal. But from this illustration, from this fist illustration, can you appreciate that it's essentially the same membrane? It's continuous. Does that make sense, what I'm trying to say? So heart is like pressed into the pericardium, okay? It is pressed into the serous membrane, so that visceral pericardium surrounds the heart, and parietal pericardium surrounds the cavity. There are other organs that would exhibit the same behavior. For instance, lungs, okay? Lungs are surrounded by pleura. Visceral pleura will cover the lungs themselves. parietal pleura will cover pleural cavity. If you will try to trace, like using a pencil for instance, a pleura here, the blue line, okay? The blue line. You will see that the line is continuous. It never ends, you know, it kind of goes around. That makes sense? Another organ that, well, it's not even one organ, many organs that are covered with serous membrane are the organs of abdominal peritoneal cavity. So organs that are covered by visceral peritoneum, for instance, transverse colon or liver, okay? Most organs in the abdomen are covered by peritoneum, okay? And of course, parietal peritoneum, which is red on this picture, okay, is covering the surface of the peritoneal cavity. Now, why I pay so much attention to visceral membranes, visceral serous membranes, and parietal serous membranes? Not so much in peritoneum, but in the lungs and in case of the heart, you can see that there's a little tiny space between the visceral and parietal layers. Can you see that space? You can, right? What do you think this space is filled with? Fluid. Yes, fluid. You don't have to raise your hand. Just don't start brawl, you know, for who said it first. Okay. So it is filled with fluid. Where does this fluid come from? Can you be more precise? But how does the organ feel? Like, how would the heart feel? Slimy. What produces that quote-unquote slime? Not the mucus, the serous membrane, right? So that fluid is essentially the secretion that serous membranes produce. Make sense? Now, go ahead.
Serous fluid in between the visceral,
layer and
the organs, that space of the fluid.
Stop for a second. Hold on for a second. You have an organ, and you have a cavity in which this organ is housed. Say heart and pericardium. Heart is covered with a serous membrane, and inner surface of the cavity is covered with a serous membrane. Okay. Between organ and the cavity, there is a space. Can you see that space? It is filled with fluid.
Okay, now I see it.
Did you get what I'm trying to say? Awesome. So this fluid essentially is a lubricant. For lungs, for lungs it's more than a lubricant. I'm not going to go into it too much. For heart, it's a lubricant. Now, what if, let's say, hypothetically, that layer, parietal layer of pericardium or visceral layer of pericardium becomes inflamed. There's some sort of an infection going on. What happens in any type of inflammation? We didn't cover it yet. It's just from your day-to-day experience. What happens in any type of inflammation? Swelling occurs. Because of what? Fluid rushes to the site of inflammation. So imagine you are a heart, and fluid starts to accumulate around you. You cannot contract as you used to contract. That is called pericarditis. If it happens in the lung and, pleura becomes inflamed, pleuritis. Fluid accumulates, lung can collapse. Okay? So, it's, it may seem like very accessory structure, but it may alter the functions of the organ, tremendously, obviously. Does that make sense? Questions? Can we move on? Now, tissues. What I hope to accomplish today, well, not I, but what I hope we accomplish today is that we cover tissues and we do at least part of the lab. You may have seen a couple of lab handouts that I uploaded. The handout number 1, which is about anatomical terminology, come on, you can do it without me. We can review it, and we will, but you don't need class time to fill it out. Tissue handout, that's what we're going to do together. You're going to take microscopes, you're going to pull them out, I'm going to pull out some of the tissues are right there. You're going to look at the slides, you're going to draw the slides, we're going to pop them up, it's going to be a lot of fun. Okay? So, there are 4 types of tissues, period. Okay? That's very important. 4 types of tissues. Nervous, muscle, epithelial, and connective. And I expect you to know all these 4 types. What is the function of, without even maybe looking at the slide, just think about it, think about the name. What is the function of nervous tissue? Send signals. Perfect. Communicate, right? Receive the stimulus and deliver it to the brain or spinal cord or whatever, and then send the response to organs, to glands, and to muscles. Is that clear? What is the function of muscle tissue? Movement. Perfect. Just one word. Whether it is a voluntary movement that is done by skeletal muscle or involuntary movement, pumping action of the heart, or changing diameter of the intestine or blood vessel. What is the function of connective tissue? It's kind of easy to remember. To connect. Perfect. To connect. Really. If you think about it, it connects other tissues. It connects parts. Connective tissue in your skin connects parts of the skin and muscles. Connective tissue, bones are connective tissue. They connect different parts of the body. They connect muscles, to each other, or muscles to the skin or whatever, okay? Connective tissue in the walls of organs connects layers in these walls. What is the function of epithelial tissue? Too broad. Boundary. Bingo. Boundary. It's a boundary. Boundaries can be external, they can be inside, but it's a boundary. Make sense? What is important, epithelial tissues, I'm trying to think if there are any exceptions to this rule, and I cannot. Every epithelial tissue is exposed to some kind of lumen, empty space. Does that make sense? And we will see examples of it. My expectations from you regarding these 4 types is that you never tell me that, say, bone is the separate type of tissue. Like if I ask you which of the following is not a tissue type, you think, okay, nervous, muscular, connective, epithelial, period, 4 types. That's it. Okay, there are subtypes, we'll talk about them, but those are 4 types of tissues. Am I clear? Cool. Now, where do these tissues come from? Well, when we are, after we stop being haploid and become diploid, become embryo, or cells, Well, first, we can become anything. You know, like your parents, when you like really young, they tell you you can become everything. That's not true. At this age, not anymore. You could become everything when you were like 2 days after conception. This point, embryo is assembly of stem cells. And these cells that comprise that first embryo. Okay. These cells are called totipotent stem cells. Well, stem because everything stems from them, and toti means total. They can become any cell in the body, literally. Totipotent stem cells can become any cell in the body. And then, now I'm going to tell you 2 words which I just found out today are not in the study guide, but you must know them. I want to be very clear about it. These 2 words are differentiation awful, and proliferation. Now you're going to tell me what these words mean. What does differentiation mean? When cells start to differentiate, does anyone know what it means? Excellent. So they become more specialized. Yes. Differentiation is when cells, you need better spelling? Was it lousy? Differentiation's okay? Cells become more specialized. What about proliferation? Yes, go ahead. Not the function. You're right about multi-part, but not the function. When cells proliferate, what does that mean? Yeah, they become more numerous. That's it. That is it. When we say cells proliferate, they become more numerous. Proliferation means, multiplication of the cells. Clear? Awesome. So this totipotent embryonic stem cells start to differentiate and proliferate, both. And first, and only, they differentiate into pluripotent embryonic stem cells. What do you think is the difference between totipotent and pluripotent? Toti means everything, right? What about pluri? Plural, what does that mean? Many, right? Many. So pluripotent stem cells can become many other tissues, but they are somewhat restricted. Is that clear? That they are restricted a little bit. So you have 3 kinds of pluripotent stem cells. Essentially, they belong to 3 types of embryonic tissues. The outer, now, for the simplicity, imagine the embryo as a sphere. Okay? The outer layer of the embryo is composed of ectoderm. Okay? So what can ectoderm become? Can become skin, so epidermis, skin glands, okay? Can become certain bones, right? But can become certain glands like pituitary gland and adrenal medulla, nervous system, and ends of your digestive system, mouth and anus. So essentially, your front end and your rear end have the same origin. Okay. Now, what do you have to know? Which parts do you have to know that are, generated? Epidermis, which means pretty much skin, okay? So let's write it down. Skin, nervous system, and two ends of digestive. Please know that these three things come from ectoderm, from the outermost layer of the embryo. Am I clear? Fantastic. Mesoderm, layer in between. Mesoderm produces connective tissues, most of the skeleton, blood, well, cartilage, pretty much, if you think about it, connective tissues, skeleton, and cartilage, sort of pretty much all connective tissue, including blood. Muscles, synovial membranes in the joints, serous membranes, kidneys, and gonadal lining. Again, organs which you have to relate to mesoderm for the exam purposes, about which I can ask you. Connective tissue, bones, and blood, muscles, kidneys. Am I explicit enough in my requirements? If you think that I'm explicit enough, blink once. If you don't think, blink twice. Okay? Go ahead. Ector's on the outside. Meso is the next. Endo is the deepest. We're getting there, okay? And then finally, endoderm. It gives rise to the organs that are sort of hidden deep inside of you, such as digestive and respiratory system, okay? So, know that digestive comes from endoderm, respiratory comes from endoderm, okay? Glands, don't worry about them. I understand it's too much. So, to kind of sum up, what you can have a question. Which of the following systems, okay, endoderm gives rise to this organ system. You have the list, you pick one. Does that make sense? So, again, for the ectoderm, skin, nervous system, and mouth and anus. For mesoderm, connective tissue, pretty much like all of it. Skeleton, connective tissue proper, cartilage, muscle, kidneys. For endoderm, respiratory and digestive system, obviously except for mouth and anus. And that's pretty much it. Is that clear? Let's go back to our stem cells. Pluripotent stem cells start to differentiate and proliferate more. They become more specialized. For instance, some of them will be specialized towards becoming lungs or structures of digestive tract or nervous cells. For instance, neuroblasts are the cells that eventually become neurons. Okay, glioblasts, the cells that become glial cells of the nervous system. Many of them establish so-called lineages. For instance, there's a myeloid lineage. Okay, so cells of a certain lineage will develop a certain organ. Cells of, say, a myeloid lineage will become blood cells. Cells of lymphoid lineage will become cells of lymphatic system. Does that make sense? Think about, I always give example of like football players. When your kid starts to play football, they don't know anything. They can be any any kid on the field, right? They can play any position at 4 years of age. And then eventually, you can see that like kid really can run. So he can play, I don't know, wide receiver or cornerback or safety. And then turns out that he's better at defense. He doesn't really catch the ball that good, but he's really good in the defense and tackling. So he becomes a defensive player. And then his size, warrants him to be cornerback rather than small safety. And, it gets more and more specialized. Same goes with the stem cells. Now, why stem cells are so cool? Why there's so much hype around them? Exactly. Why it's so important?
Cloning or replicating organs.
Yes. So cloning, I wouldn't touch this with a 6-foot pole, but yeah, you can pretty much make any organ, right? Now, what is the main source of stem cells? Yes, well, not babies, embryos. So it's like very early stage abortion material. I'm not talking about ethical component of this, but biological component. One of the problems is that there can be issues with immunological rejections. Does that make sense? So we have our own tissues, and if we receive stem cells from embryonic source that is not related to us, there can be significant immunological differences between cells from that embryo and us, and our immune system can recognize those embryonic stem cells as foreign and can technically attack them and cause the rejection. It can be mitigated by taking immunosuppressants, but it's a problem, right? On the other hand, if you choosing one, either dying from myocardial infarction or heart failure, or taking immunosuppressants for the rest of your life, which will happen. I take immunosuppressants any day of the year, okay? And now, ethical concern, of course, that it's an abortion material. And in 2012, 2 American scientists one of them from, I think both of them from University of California, San Francisco.
Are you talking about the plasmapheresis?
No, but you will see this is what I'm talking about here. Yamanaka and Gordon, they received Nobel Prize in 2012 for the work that they have done in 90s and 2000s. They developed a method to produce inducible pluripotent stem cells, which is pretty cool. I never saw the movie, the story of Benjamin Button, whatever story it is, but he kind of goes back in time, right? So he was born like an old guy and then becomes younger and younger. Inducible pluripotent stem cells are essentially Benjamin's buttons of the cellular world. You take a normal differentiated cell from the human body. Let's say the easiest one to take, say, fibroblast from the skin. Okay? It's specialized cell already. You take this cell and treat it with certain chemicals like proteins and growth factors, and it starts to go backwards. It becomes de-differentiated. So you run this process that you see here outlined, the stem cell development, you run it backwards. It doesn't go all the way back to totipotent, but it becomes pluripotent stem cell. This is the coolest thing, honestly. So imagine you have a patient who has, I don't know, a stroke. Okay? Well, I don't know how it can can it treat stroke, but let's hypothesize. Okay, patient who has a kidney failure, or liver failure, whatever. You take that fibroblast, treat it, and it becomes pluripotent stem cell. And then, using certain chemical cues, you direct development of that stem cell into the liver or into the kidney. Essentially, well, technically you can grow the new organ in the lab. And what is important, this organ is going to be the same immunologically as the future recipient of the organ. So you become the daughter and the recipient at the same time. Does that make sense? So there is no immunological conflict. There is no rejection. And it's pretty cool. Okay? You can, technically speaking, you can take that stem cell, for instance, if somebody has a blood cancer, leukemia, or another sort of blood problem, you can take stem cells, de-differentiate them, make them pluripotent, then completely obliterate bone marrow, and direct those stem cells to become myeloid cells, to become bone marrow cells, populate bone marrow, and the person has normal blood now. This technique is still in the works because there are some issues with those stem cells. They aren't as good as embryonic because they they have that, how to say, so when you say 34-year-old, those stem cells are kind of carrying information that you are old. Does that make sense? They aren't as good as embryonic. They're not fresh. Okay? But one day, I believe, we're going to be able to grow livers and kidneys and muscles and I don't know about brains, but, you know, all types of organs and replace them, hopefully. So we'll extend the quality of living. Yes. Not the heart. Well, we can do it, but what we use from pigs are the valves, the cardiac valves, yes, because they are not extensively vascularized and it's easy to transplant pig valve into human. There's practically no rejection. No, it's because it's just a valve. It's pretty much like a cartilage. So, well, it's not cartilage, but connective tissue. It's not a lot of blood vessels there, so you're pretty safe. Blood, we cannot take blood from pigs. No, no way. Now, 4 types of tissues, and we're going to start with epithelial tissue. It's a barrier, right? So it lines surfaces. Which surfaces can you think of? Which surfaces do we have? Skin. That's epithelial tissue. What else? It's not surface. It has to be, when we talk about surface, it has to be, you know, kind of exposed to something kind of sort of empty. Huh? Nails here. It's all pretty much, it's all skin, just different kind of skin that is changed somewhat. Come on. Digestive system. The entire digestive system is the surface, right? It faces the lumen. Stomach, esophagus, intestines. What else? Parts of the reproductive system. Essentially, all the tubing of reproductive system is covered in epithelium. Speaking in general, any tubes in the human body, and I always say, you know, human body is an amazing example of plumbing. Okay, we have a lot of plumbing. All these tubes are covered with epithelial cells of different sorts. Okay, blood vessels, parts of the renal system, digestive system, respiratory system, any ducts that can be found in endocrine system and exocrine glands, anywhere. It's epithelial cells. Now, the classification of epithelial cells is based on two things. First, the number of layers. Second, the form of the cells. Okay, the shape. Number of layers is pretty simple. You have simple, one layer, and you have stratified, many layers. See the difference? Now, shape-wise, you have squamous, which are flat, cuboidal, which are surprisingly cuboidal, and columnar. Well, columnar in shape. There are few, well, I want you to know two more types, just slightly different shape-wise and arrangement-wise. One type is pseudostratified, and another one which I will show you a little later because we're going to talk about each type separately. Another type is transitional, okay? And I will explicitly tell you which types you must know and what you have to know about each type of the tissue, okay? Now, cells, epithelial cells, since they represent the barriers, okay, they are connected by so-called junctions, okay? Junctions can be of three different types. One is tight, okay? So tight junction prevents any sort of transport across the layer of epithelial cells. So you can see the two membranes of the epithelial cells, and they are somewhat stitched by the tight junctions. So essentially, these junctions prevent anything from going from one side of epithelial barrier to another. Does that make sense? Think about it. Think about tight junctions as helping epithelial cells to form a perfect wall. You can find the tight junctions connecting epithelial cells that line blood vessels of the brain. If you would look at the blood vessel, on the inside, the part that faces the lumen is of course lined with epithelial cells. Those are nuclei. Does that make sense what I'm picturing here? It's the cross section of the blood vessel. Okay? So this is lumen, and those are epithelial cells. Okay? These cells are connected in the brain. Cells are connected to each other by the tight junctions. Why do you think it is so important to establish such a tight barrier between the blood in the brain blood vessels and the brain tissue itself? You shouldn't. It's, well, if the blood vessel ruptures, then you're boned anyway. I mean, those tight junctions are not going to help you. Exactly. So you don't want plasma to leak out into the brain. There's another reason. Blood is... Go ahead. Not so much, not junctions. You established a complete tight barrier. If something is in the blood, Oh, oxygen will go back and forth, no problem. We need oxygen to the body. Different types of, well, big molecules or even microorganisms, so they cannot reach into the brain. Think about it. If we have a kidney infection, it's unpleasant, but, you know, we can deal with that. What I mean is, like, if kidney function is compromised and then is restored back, it's okay. Okay. If brain function is compromised, we boned. Brain is precious. We need to protect it. That's why we build such a tight wall around the blood that can carry all kind of crap, to prevent all that kind of crap from reaching into the brain. Does that make sense? Anywhere you want to prevent the transport of things from the lumen into the surrounding tissue, you're going to find tight junctions. Pretty much the opposite of it is gap junctions. The gap junctions are essentially the channels through which ions can move from one cell to another. Make sense to you? Okay? They establish communication between the cells. So for instance, if I want to tell someone just in classroom what's going on, all I need to do is to go to the door, open it, and scream, right? That's sort of an example of the gap junction. Okay? I don't need to, like, walk around and whatnot. Anchoring junctions, which you can see right here, their function is to just hold epithelial cells together. There are several types. The desmosomes, junction adherents, and hemidesmosomes. So desmosomes and hemidesmosomes, they keep cells together. They prevent epithelial layer from falling apart. Does that make sense? So, for instance, desmosomes hold the lateral surfaces of the cells together. Okay? It's like people holding hands and holding them pretty tight. Okay? Hemidesmosome is called hemidesmosome because cell is only one side of the junction, if you know what I mean. If you look at it, let me erase the drawing here. If you look at it, you can see that in case of hemidesmosome, it attaches the cell to the basement membrane, sort of attaches bricks to the foundation. Does that make sense? So desmosomes attach bricks between each other. Hemidesmosome attaches the brick to the foundation, the epithelial cells to the basement membrane. Okay? Every epithelial layer has a foundation, the basement membrane. Okay?
So desmosomes and hemidesmosomes are part of every junction?
No, they are anchoring junctions. Okay, let me explain. So say if people want to make like a wall, nobody can penetrate. They will probably hold each other like this, right? Very, very tight. That's tight junction. I'm going to skip gap junctions. It's like different story, okay? It's tight junction. If we just want to make like a row of people, we're going to hold our hands. Make sense? That's going to be desmosome. Just hold each other's hands. The role of hemidesmosome, and I cannot give you any good analogy here, you have a layer of epithelial cells, and this layer is attached always to the basement membrane. Like bricks of the house are attached to the basement, foundation. That makes sense? So that junction that attaches the lowest layer of the bricks to the foundation is called hemidesmosome. That's the only difference. They are not very tight. They're sort of leaky, but they hold epithelial cells together pretty well. Did I answer your question? If not, tell me. We can I can clarify during the break if it takes a little bit longer. Okay? Huh? It's fine? Okay. Now, junction adherence, since it contains the actin, it allows epithelial cells to move a little bit. Does that make sense? Think about your skin. Your skin is being stretched all the time. And cells that comprise your skin have to have a certain level of freedom to move around. Adherence junctions allow them to move around. It's like not holding hands directly, but if I hold a rubber band and Jack holding rubber band, we can move around a little. We're not going to fall apart, but we have certain degree of freedom. Does that make sense? Okay. Understand the functional differences between gap, tight, and anchoring junctions. What do they do? Okay? Now, we're going to talk about individual types of tissues. First, we're going to start with the simple epithelium, and we're going to talk about simple squamous. Cells are fairly flat, thin, that's kind of it. I don't know what to say, what else to say. They're flat and thin, okay? So if you think about them forming a barrier, that's going to be sort of a lousy barrier, very penetrable. Not surprising that you're going to find this type of epithelium in the places where a lot of diffusion, filtration, any other sort of transport occurs. Does that make sense? Now, let's find out altogether where you're going to find this kind of transport based on your personal experience, just with humans. Lungs. Gas exchange. Does that make sense to you? There's incredible intensity of transport in the lungs. So you're going to find them in the alveoli, they call them air sacs on this picture. Alveoli of the lungs. In fact, that's the cross-section of many individual alveoli. So that's alveolus. That's another wall of alveoli. That's alveoli. Does that make sense? Okay. Squamous, simple squamous epithelium will line all the blood vessels. That picture that I showed you here, this is the simple squamous epithelium. Cool? Okay. Blood vessels, lymphatic vessels are the same. Lymphatic vessels are very similar. Okay. All serous membranes. Pericardium, pericardial membrane, pleural membrane, peritoneum, all composed of simple squamous epithelium. Now, since we have not covered kidneys, I'm going to introduce it to you, but I'm not going to ask this, I promise. What is the function of kidneys? Remind me. Filter, right? So, filtration happens in the glomeruli. Honestly, I wouldn't call it much of a filtration. It's more of a dumping, a bunch of stuff. Essentially, what happens in your kidneys, that's first step of production of urine, is that liquid, plasma of your blood, is being filtered. Water, ions, small organic molecules are all gone. Proteins and cells remain in the blood. And that is a very intensive process. There's a lot of filtration occurs. To give you an idea how much your kidneys filter a day, they filter 120 liters of fluid. If that doesn't ring a bell, this is more than 30 gallons, like 35 gallons of fluid filtered by your kidneys every day. That's a lot. And that happens through the simple squamous epithelium in the glomeruli in the kidneys. Does that make sense? So simple squamous, filtration, transport, diffusion, anything that involves movement of things. Yes? Good question. No. And if you give me like 5-7 minutes, you will see, I will explain, we'll get to digestive, and I will tell you it's slightly different. Okay? Now, since the question about, we have a question about digestive system here. If you think about it, blood is pretty clean. Air is not really aggressive. So, on the other hand, digestive system involves a lot of sometimes pretty mechanically crude material. Do you see what I'm trying to say? Like if you ever vomited, and I bet everybody in this room vomited at least once in their lives, you know that it looks pretty crude. It's not like clear fluid, okay? So, simple squamous epithelium will not survive that type of mechanical assault. Cuboidal. Cuboidal epithelium deals with secretion and absorption. Okay? Well, cuboidal. I promise it's just a reminder. I didn't turn off the sound, my bad, but phone is off. Didn't turn off the sound now. Where are you going to find cuboidal epithelium? Kidneys, tubules of the kidneys. There's a lot of absorption and a lot of secretion. Glands, many different glands, like thyroid gland. That's the picture of kidney tubule and thyroid gland. You're going to find cuboidal cells too. Ovaries, a lot of secretion in ovaries. Okay. Again, simple cuboidal isn't very good in withstanding mechanical assault. Okay. And here we come to Cody's question about digestive system. In digestive system, you're going to find simple columnar epithelium. You can see that the cells of simple columnar epithelium are extended. So epithelium is rather thick. You see that on that micrograph? It's rather thick. Sometimes it's ciliated, well, whatever. Practically all layers of simple columnar epithelium, all locations, produce mucus. And we'll talk about mucus. Now, the function of columnar epithelium, for instance in digestive system, is absorption. Here's the thing, going back to your question. When we look at simple squamous, air gets into the lungs. Nothing happens to it. It just, diffuses into the blood right away. Blood gets to the glomeruli, and water and ions just directly diffuse, filter through the simple squamous epithelium of glomeruli. However, if you will think about your intestines, what do you eat? Give me examples. Salad. That's it? Everybody eats salad, like goats? Pizza, meat, okay? Does pizza consist of little pizza molecules? Salad of little salad molecules? No? No? What are the three nutrients, main nutrients that you can find in any packaging, written numbers? Proteins, carbohydrates, those carbohydrates. Those carbohydrates. Huh? They're not energy bearing. Fat. Okay? Fat, carbs, proteins. Make sense? So when you eat, say, pizza, after certain steps that pizza goes through in digestive system, you end up with a bunch of proteins, fats, and carbohydrates in your intestine. That is not what is being absorbed in the intestine. Those fats, those lipids, carbohydrates, and proteins are being digested further to the elementary building blocks like amino acids or fatty acids or monoglycerides or simple sugars to be absorbed into the columnar epithelium. So you see, it undergoes certain breakdown. It's not just direct absorption. Okay? So it's going to be columnar in the digestive. Also, simple columnar epithelium bears so-called goblet cells. Okay? These goblet cells contain vesicles. You see this? These vesicles here. These vesicles carry mucus, consisting mostly of the protein called mucin. Okay? So, mucous membranes, like the membranes covering your stomach or intestines, they receive mucus from those goblet cells. Mucous membrane that covers your respiratory passages, mucus is produced by goblet cells. Does that make sense? Terrific. Now we're going to find them, well, digestive tract, non-ciliated, gallbladder, okay, bronchi, ciliated columnar epithelium. Now, what is the function of cilia on the cells? Anyway, what do we need cilia for? They propel things. Now, if you think about it, in digestive system, propulsion occurs because different parts of digestive system contract and dilate. Right? In your bronchi, that cilia on the surface of columnar epithelium, what does it propel? What do you think? What covers the epithelium in your bronchi? Mucus. So that cilia will propel mucus. Where does it propel it to? Yes, and then, well, yes, throat, and then, what do you do with it? Oh, we would be like knee deep in the mucus then, if you would all spit out. You swallow it. Okay, because this mucus contains crap that you breathe in. Make sense? So you will see cilia in the places where you need to move stuff, like mucus in your respiratory system. Or you will see ciliated columnar epithelium in the uterine tubes. What does cilia move there? In the uterine or fallopian tubes? Eggs, right? That makes sense? So columnar epithelium will produce a lot of mucus, will absorb, sometimes, not always. There's nothing to absorb in uterine tubes, for instance. There's not really anything to absorb in the respiratory system. Okay? It's a lot of mucus secretion. Make sense? Pseudostratified. It's also called pseudostratified columnar. Now, why it is called pseudostratified? It looks like stratified, but it's not. Okay? If you would look at this micrograph, you will notice there's pretty much like it's one layer. Okay? There are no multiple layers. But cells in this layer are arranged in a way that nuclei of these cells give you an impression of more than one layer. Does that make sense? Can you see that? So I have layer 1, layer 2, and maybe layer 3. Okay? So, in fact, it is one layer of cells, but the arrangement shows as many. Make sense to you? Or not? That is why it is called pseudostratified. Essentially, columnar cells arranged in an awkward way. Okay. Also, mucus. It's pretty much similar to columnar mucus secretion. If they are ciliated, propulsion of mucus, for instance, in a trachea, you have pseudostratified ciliated epithelium that moves mucus up. In the ducts of the male reproductive system, cilia will, move the sperm cells towards ejaculatory duct and then eventually urethra. Okay, from testis through the epididymis, vas deferens, towards the ejaculatory duct. That makes sense? Sure.
Why not produce? Is it a way that we can produce or not produce as much mucus? Since mucus holds a lot of bad things.
Okay.
It is one of the sources of cancer.
Wait. Slow down. Start up. So, what is your start over so I can split the question and introduction? Go ahead.
I know. So we produce mucus is like a protective type of source.
Of course.
Why is it a way that we can not produce as much mucus, or is it somehow that we can limit mucus production?
Voluntarily, no. Mucus is normally produced by your, say, goblet cells in the columnar epithelium. It is not produced at the constant level all the time. For example, if you have inflammation, if you have nasal infection, it means that you have some kind of bugs in your nose. And mucus cells, goblet cells, will start producing mucus at the higher level in a hope to catch them up, immobilize them, prevent them from spreading, so that then you can, snort them out. Make sense? If you have sort of an irritation in the trachea, mucus production will be up. Because if it's an irritant, it may be dust. We need to capture dust from getting it into the lungs, so mucus goes up. In some diseases, mucus is produced at the levels that are abnormal, like in cystic fibrosis. People produce abnormally high level amounts of mucus, which clogs up pretty much all of the ducts. It's not only the most commonly known symptom of cystic fibrosis is the obstruction of respiratory passages, accumulation of mucus in the lungs, in the bronchi, and the subsequent infection. But it also obstructs the ducts of pancreatic gland, preventing people from digesting food normally. But mucus, we cannot regulate mucus overall, not voluntarily. And I can think of maybe drugs that can sort of decrease its release systemically, like epinephrine, phenylephrine that you take when your nose is clogged, that will reduce mucus production. Does that make sense? As a part of response. But it is not very targeted response. Did I answer your question? If you have, we can chat. Yes.
Makes us produce more mucus.
Well, certain stimuli, yeah, they will increase the mucus production, of course, yes.
So we can decrease that stimuli that we won't be producing as much.
Yes. If we breathe clean air, the amount of mucus that's produced in the respiratory tract will go to the sort of a basal. Stratified squamous. Now, this is so we've got 4 simple layers, right? Simple squamous, simple cuboidal, simple columnar, and pseudostratified columnar. Now, this is the stratified epithelium, which means it contains more than 1 layer of cells. And if you look at it, look at the micrograph. I'm going to show you the layers. This is the layer 1. This layer 2, 3, and this is 4. Okay? Does that make sense? I don't know why this black thing doesn't go away. Okay, so you have multiple layers. You can see them. This part is definitely different from this part. Right? If you would look at the bottom layer, like deeper layer, these cells look more not as flat as the layers. These cells will look not as flat as these cells. The basal layer of stratified squamous epithelium looks more like cuboidal, and then they become more and more flattened. Does that make sense? Now, another interesting feature about any squamous, stratified squamous epithelium is that not all the cells in that epithelial layer are alive. Only basal cells are alive. They constantly divide and produce new cells, which then migrate upwards. And while they migrate upwards, they die. So essentially the external layer, the superficial layer of stratified squamous epithelium is dead. Okay? Now, this dead layer is the perfect protection because you don't really care about the dead cells. You can lose as many as you want. Does that make sense? It's like forest. Forest, trees in the forest are living. They're new trees, growing every year. And then we cut them and we build, fences, houses, enclosures. They are built of dead trees, right? But new trees, we cut the trees and new trees will start to popping up. Okay? Same goes with stratified squamous epithelium. Outer cells will be always, we will always lose them. Like cells of the skin, we will always shed the skin. You can find like enormous amount of skin in the world. If you would check the bacteria that are present on things like laptops, cell phones, folders, anything that you handle, gym equipment, your car keys, your car steering wheel. You know what's going to be the most common bacteria? Your skin bacteria. Whatever's found in your skin, it's probably the most common bacteria that you can find in human environment, in a built environment. Okay? We'll lose these cells all the time because the basal level keeps producing them. And it's obviously protection, and not just protection, it's really good protection. Protection against mechanical forces, abrasion. Skin, obviously. A lot of insult, a lot of abrasion. Where else? Where else we can find a lot of abrasion? It's pretty much all skin. I mean, those are skin structures. They derive from skin. Parts, yes. Oropharynx and laryngopharynx, yes. Throat. Mouth, yes. Front end and rear end. Anus, of course. Not the entire thing, but closer to the anal orifice. Absolutely. Reproductive organs, especially vagina. A lot of rubbing happens there. Okay? Seriously, it is exposed to the outside world, and there's abrasion force, so it's simple. It's stratified squamous. Okay? Everywhere you think about abrasion forces, there you go. Some of the stratified squamous epithelium is keratinized. We will talk about it. That's skin. Everything else is non-keratinized. So, say, stratified squamous epithelium that you can find in your gums is non-keratinized. Same goes for anus. Same goes for vagina. It's non-keratinized. But it's same concept of multiple layers. Okay? Transitional. Kind of funny. It is stratified epithelium, okay? It is. And if you look at it, it's sort of, more of, oval cells, like extended cells, deeper, and then become more and more cuboidal to almost squamous at the superficial layer. So it looks a little like stratified squamous, but the difference is all cells are alive. Does that make sense? You don't have, in transitional epithelium, you don't have a clearly defined layers. Can you see the lack of clearly defined layers? Okay? I mean, there are layers, but they aren't really different. You can find transitional epithelium covering the walls of the bladder because it is stretchable. Okay? For instance, everything that's stretchable, like ureters, they stretch a lot. Bladder stretches a lot. Parts of urethra stretch. Okay? So that's where you're going to find transitional one. Now, before we move to exocrine glands, there's 6 types of epithelium. Is what I will ask you on the exam. So I have 4 simple types: squamous, cuboidal, columnar, pseudostratified. And you have transitional, and you have stratified squamous. Only those types will be on the exam. I absolutely guarantee that. You have to understand the function, and you have to understand where they can be found. I promise I will not ask you really obscure, little tiny locations. Questions will be pretty obvious, okay? If I ask you about, say, stratified squamous, you can imagine it's going to be something very obviously abrasive, skin or mouth, okay? If I ask you about cuboidal, I focused on thyroid gland and kidneys, okay? So have that appreciation, okay? It can be a question, this layer, this type of epithelial tissue produces a ton of mucus and can absorb nutrients. Simple column, okay? Make sense? So that type of question. Don't worry, when I give you a quiz, you will see what it's going to look like. The questions, okay? Glands. We'll we will definitely go about the glands, and then we're going to take a break. Okay, glands, 2 types, exo and endocrine. What does exo and endo means? Which one is inside? Endo, and exo is outside. So endocrine glands, where do they secrete? Oh shit, it's on the slide. It's boring. Endocrine glands, well, blood, right? Exocrine glands secrete outside. Give me an example of the outside. That's it? That's it? Sweat, but what else? Where else can exocrine glands secrete? It's not technically an exocrine gland. It's a mucus cells, huh? Okay. Yeah. But why are you so hesitant to get deeper in the human body? Is there any outside in the human body? Outside in the human body. Okay, saliva. Excellent. Don't hesitate. Go deeper. Respiratory system. There are some exocrine glands, yes. The entire digestive system. That's the whole in your body, right? So the point is, exocrine glands produce whatever that goes not into the blood. Does that make sense? For instance, your pancreatic gland, when it produces pancreatic juice that enters your intestines, functions as exocrine gland. When the same pancreatic gland, pancreas, produces insulin, which gets directly into the blood, it is endocrine. You see the difference? Can you see the difference? Okay? Exocrine glands do not have to produce whatever they produce on the skin or on the eyes. Okay? It can be your digestive system. It can be the tubes of your reproductive system. It's still going to be exocrine secretion. Does that make sense? Okay? Now, if you would look at the glands on the left, you will see that there are, first of all, there are several types. Major types, alveolar and tubular. Alveolar glands have this little circle-shaped sac at the end. Tubular don't. Okay? Glands can be branched or not branched, like this simple tubular or this simple alveolar. Are you keeping up with me so far? Awesome. Glands can be compound when you have, say, branched alveolar glands and then branch further. So you have second and third order of branching. Does that make sense? Now, I do not require to know which gland represents which structural type. All you have to know that there are tubular and alveolar glands in terms of exocrine. And what does the compound mean? What does the branched mean? I believe it's pretty fair and pretty simple. Now, exocrine glands, based on the mode of secretion, can be divided into three types: merocrine, apocrine, and holocrine. Merocrine glands package their secretion into the little vesicles that they release from the cell. Does that make sense? Whatever is secreted is neatly packaged in the vesicles and is released from the cell of the gland remains intact. You see, nothing happens to this cell. In case of apocrine glands secrete by pinching off the fragment of the cell. In apocrine glands, cells do not remain intact, but they are not dying completely. In case of holocrine glands, cells die. They die, they explode and release their content, and that's the secretion. Okay? The examples, I bet you want examples. Regular sweat, like the sweat that we produce forehead, okay, or on the arms. That's the merocrine sweat. And let's be honest, you know, that sweat that's on your face, for instance, it doesn't stink. Seriously, I mean, it's salty a little bit because of the salt, but other than that, it doesn't stink. On the other hand, sweat that is produced by the glands in your armpits when you're growing, it's the apocrine sweat, stinky one. So fragments of the cells are pinched off. These fragments contain not only water, not only salt, they also contain a bunch of organic molecules. Does that make sense? That bunch of organic molecules that is metabolized by skin bacteria is what produces that sweat. That smell, sorry. And finally, holocrine glands, sebaceous glands. That's what Mar mentioned. They produce sebum. Cells die and release all their content. See the difference? Please know there's 3 types of exocrine glands and know the difference between endocrine and exocrine glands. I'm going to ask you a question now. You see the glands here, you know, on the left, in this large picture. They folded. Why? Why glands are folded? Even, huh? So again? Well, no, you actually need secretion out. They folded. Why? Why anything is folded in a human body? What?
It's like all one piece. It's like one structure. It's not like segments necessarily.
Packaging so it can fit.
We're getting closer. There you go. Who said it? Good job. This is one of the things that, I mean, if, I don't know, during the AP1, we're going to run into it on many occasions. When you take AP2, you will run into it on many occasions. Something is folded. More surface area. Make sense? Got it? Okay. Let's take a break.